Hello and welcome to this video, examining the situation in which a Chinese scientist claims to have created Crispr Babies by editing the genome of twins. This was to make them less susceptible to HIV, and it has been described as eugenics by some more critical members of society. Whilst Mengele may have seen this in some great light, both the scientist's Chinese peers and those across the world have seen it as something less than ideal and have in fact slammed it. The university that he is based in says it is investigating the matter immediately, particularly in light of the fact that this associate professor announced their findings in a YouTube video.
Two beautiful little Chinese girls named Lulu and Nana came crying into the world as healthy as any other babies a few weeks ago. The girls are home now with their mom Grace and dad Mark. Grace started her pregnancy by regular IVF, with one difference. Right after we sent her husband's sperm into her egg, we also sent in a little bit of protein and instruction for a gene surgery.
Rather than a more appropriate avenue, such as academic publishing, this story goes beyond the simple fact that there is a rogue scientist conducting unapproved research. He is using modern technology that is yet to be proven in the role it is being applied to. CRISPR has been tested and is currently being tested in conditions such as Duchenne muscular dystrophy, and you'll find a video to that linked in the top right. But this is a clinical trial. This is going to affect adults who have an established genome, something that will not change randomly, unless of course they develop a pathology such as cancer, but that is a very limited situation. In this case, the scientist has taken their research and applied it to developing embryos. This has then led to children being born who are genetically modified, designer babies, and the newer term CRISPR babies. This is a violation of any number of academic ethics and standards, and the university that he is based in has decided to treat it as just that. Their 5-part video series only compounds the situation and provides evidence to that investigation. The truly disconcerting aspect to this is that the scientist feels that what he's doing is right.
Family is a society's bedrock. Our children are at the center of family life. If we can protect a little girl or boy from a certain disease, if we can help more loving couples start families, gene surgery is wholesome development for medicine. But every advance comes with a responsibility. For the last 4 decades, culture, ethics, regulation have developed entitled with in vitro fertilization technology. As a result, initial worries of eugenics have not transpired.
And that may be true, but it is certainly unethical and has certainly not been approved. In the context of this research, he has enrolled 8 families, or to-be families, with HIV-positive parents, in this case the father. They have taken the embryos and they have genetically modified them to make them HIV resistant. They have then implanted these embryos, and the children, according to this research, have been born healthy. As said, the scientific community, both within China and across the world, is furious at these actions, despite the lofty goals of this project. The problem is that the integrity of science is now being questioned. There is a process to these things that should be followed, and this was not done. For now, let's ignore the fact that this was simply a human clinical trial without the proper ethical process followed. Let's examine the technology used, CRISPR. This gene editing technology is relatively new. Its application in other diseases has been theorized. There are limited clinical trials using this technology in fully grown humans. There is no trial data showing this technology taking a fetus to birth. There is some evidence showing that it could be used in embryos successfully, but none of these embryos were ever intended to be born. They were never going to become living beings in the same way these children have. And the problem is the missed target effect. CRISPR has been shown to have some very controversial problems in its targeting and accuracy, and that it can prospectively increase complications in these children. The problem is it gets worse. This very action stems from the same ideas of eugenics seen in the 1940s, and through up until the 1980s in some places, where those with low IQ, birth defects, or other conditions and personal characteristics were significantly altered or removed from the gene pool, and often not voluntarily. This leads to significant questions about this study, where somebody has gone and drastically altered a child's genome without necessarily the informed consent of the parents, and certainly not with ethical approval. For some, this is a question of unnecessary medical procedures. For others, it is a matter of eugenics. The scientist has defended his actions through claims that the lab went through ethical approvals process, but that is largely their own internal ethical dilemma. The external ethics approvals look to have been far more cursory and far more streamlined than would be appropriate in this situation. Some have instead claimed that this situation is very much like that of the birth of Louise Brown, the first baby created through IVF. Unlike IVF, this is not just giving birth to a baby, but drastically modifying that baby in a way that is invasive and carrying not insignificant risk. The university involved has gone into damage control mode, otherwise known as arse covering, claiming that the work was conducted elsewhere. Yes, elsewhere. That is the university that they believe he did this in. Of course, it would never be claimed that somebody who was on unpaid leave had accessed their facilities, used their equipment, and done the work on site. It had to, of course, been elsewhere. Of course, all of this is being released now in preparation for the second International Summit on Human Gene Editing, which will occur in Hong Kong in very short order. This is the best possible time for these results to be released. Not soon enough for anyone to know of long-term impacts, but short-term enough that he can demonstrate that it has been a success. Of course, nobody has seen any peer-reviewed information on this, because it does not exist. He claims to intend to publish in academic journals, but that is yet to occur. He claims they'll be able to decide on whether or not the process was appropriate, but again, that has not happened, and all before one of the biggest summits. They'll be able to give direction on how appropriate and how ethical CRISPR is in the use for embryos. There are further criticisms beyond the simple ethical problems. That is that the process that was being used to genetically modify the embryo to prevent HIV infection is unnecessary. There are current laboratory-proven methods that remove HIV risk, such as removing HIV from sperm before the egg is inseminated. And these are established protocols. That makes this entire genome editing experiment redundant, as it has more severe implications elsewhere. All of this is to say, CRISPR is far too early in its development to be used in this fashion, within a laboratory, within research, within animal research. There are inherent risks that match the use of CRISPR. Within the role of embryo development, within IVF, but not within the genomic modification of a fetus. that once it is born, will become a living guinea pig for the remainder of its life. If not its life, then at least its formative years. And who knows what damage that could do to a child. Ultimately, CRISPR may go into this role. We may find the risks involved, we may find ways to mitigate them. There may in fact be means of using CRISPR as the very surgical tool this researcher describes it as, but we are not there yet, and this is far too premature. In fact, there are even legal limitations from 2003 in which China prohibits the transfer of genetically modified embryos to start a pregnancy. There are some questions as to the validity of this research. Some question whether or not it has actually occurred, others whether or not it could be reproduced. This is very reminiscent of the claims to have cloned cells from South Korea and the work of Dolly the sheep. In one case it was found to be completely false. In the other it was factual but significantly flawed. Only the future will tell fact from fiction. As a result, this research has not only tarnished science overall, it has diminished confidence in a growing technology, and ultimately, prospectively broken the laws in the very country in which the research was conducted. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.